Assalamualaikum and hi everyone. Today we'll talk about the principle of Sanger sequencing. Sanger sequencing is based on dideoxynucleotide triphosphate, or we call it ddNTP. Before we dive deeper into the principle of Sanger sequencing, let's have a look into what is nucleotide first. Nucleotide is the basic building blocks of DNA and RNA. Each nucleotide consists of a phosphate group, a pentose sugar, and nitrogenous bases, whether it's adenine, guanine, cytosine, or thymine for DNA or uracil for RNA. Without the phosphate, it is called nucleoside. Now, when the nucleotide lacks the hydroxyl group at the 2 position of the ribose sugar, it is called deoxynucleotide. Deoxynucleotide is used as a substrate during DNA polymerization. In DNA polymerization, the NTP is added to the growing chain of DNA, and this process is catalyzed by DNA polymerase. During the process, the 3N hydroxyl group of the chain attacks the alpha phosphate link of the incoming DNTP. This reaction is called nucleophilic attack. It removes the hydrogen from the hydroxyl group, and this process results in DNA elongation. In 1977, Frederick Sanger, a British biochemist, had a breakthrough idea. He thought, what if we remove the hydroxyl group at the 3N position here, so there won't be any reaction? So, the removal of the hydroxyl group here leaves only the hydrogen at the 3N position of the deoxyribose sugar. Now, the ribose sugar has 2 deoxy, hence the name dideoxynucleotide. In the classic Sanger sequencing method, there are 4 reaction tubes. Each tube contains the unknown sequence, or we call it template, primer, DNA polymerase, a mix of 4 ddNTPs for elongation of the sequence, and lastly, each tube has 1 radiolabeled dideoxynucleotide at a very low concentration. In this example, ddATP is in the tube 1, ddTTP is in the tube 2, ddGTP is in tube 3, and ddCTP is in tube 4. Now, what is happening in each tube? We look at the example here of tube 1 that contains ddATP. When the primers initiate the first binding, DNA polymerase starts the DNA synthesis as usual by adding complementary ddNTP to the template. Now, when the dideoxynucleotide is incorporated randomly into the sequence, the reaction is terminated since there is no hydroxyl group. The whole process produces DNA fragments of varying lengths in each tube. Now, based on the location of the ddNTP, for example, in this case adenine, the length of the fragment can be determined. For example, here, fragment 1 has the ddATP terminates the reaction at the 3rd base, whilst fragment 2 has the termination at the 9th base, and fragment 3 is at the 5th base. To obtain the length of the fragment, polyacrylamide gel electrophoresis can be done. Through this process, electricity is applied and migrates the DNA due to the negative charge of the DNA. In the gel, the shorter fragments travel faster, whilst the longer fragments travel slower, resulting in separation. Now, to view the separation of the DNA fragments, we use autoradiography. Since the dideoxynucleotide is labeled with radioactive compound, we can see the location of each fragment. Once we obtain the autoradiogram, we can have the sequence information of the DNA fragment based on the 4 different tubes we had for each dideoxynucleotide. In this example, reading the gel from bottom to the top shows the sequence The first nucleotide is T, followed by G, A, T, A, C, 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 A, and finally G. Now, modern Sanger sequencing uses fluorescence instead of radiolabeled nucleotides for several advantages, primarily stemming from its ability to simplify the process and automate the sequence detection. By using unique fluorescent dyes that emit distinct colors, each dideoxynucleotide can be labeled differently. In this example, the green fluorescent is incorporated into ddATP, the red fluorescent into ddTTP, the black for ddGTP, and the blue for ddCTP. Since each ddNTP is associated with different fluorescent, this allows for a single tube sequencing reaction as well as visualization through UV light. This process simplifies the procedure compared to the original radioactive method, which required separate reaction for each ddNTP. Fluorescent labeling also eliminates the safety concerns associated with radioactive isotopes, while also streamlines the process for greater efficiency. The improved method is the automated Sanger sequencing, which is widely being used currently, whereby in this method, the fluorescent labeled fragments are channeled into a narrow, thin capillary electrophoresis to separate the DNA fragments. A laser is used to detect the fluorescent dyes, and the data are processed through a software to produce a chromatogram. This automated process emerged as a more efficient alternative in comparison to the gel electrophoresis, which is more time-consuming and labor-intensive. That's how Sanger sequencing turned termination into revelation. Until next time, and thank you for watching. Assalamualaikum.